Hello and welcome to this Bioprocess International Ask the Expert webcast. I'm your host, Leah Rosen, the online editor for Bioprocess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the chat window on your screen at any time. After the presentation, we'll begin the question and answer portion, and I will ask our speaker your questions from the chat window. Your questions in the chat window will only be visible to myself and our speaker. So thank you for joining us today. It is now my pleasure to introduce our speaker, Thomas Page from Fujifilm Diosynth Biotechnologies.
Good morning, everybody. Thanks for joining the call. We're very excited and happy to share a brief overview of some of the challenges and thoughts in this area of manufacturing. So our title is a key consideration for advanced therapies manufacturing. So I'm the VP of Engineering and Asset Development here at Fuji Diosynth in Texas. Just a quick commercial for us to get out of the way. So we have we're a CDMO, and we cover a broad area of manufacturing from microbial to mammalian to viral and gene therapy. We have three main sites. The site here in Texas is the site that is a center of excellence for the advanced therapies manufacturing, and that's the one we're going to focus mostly on today. So we have commercial licenses, and we basically span all the way from PD through commercial manufacturing. So in our clinical journey in particular, we're set up down here for the advanced therapies. So we set out some years ago to make sure that we could cover the path from development through clinical manufacturing into process validation into commercial manufacturing. And we're set up for both drug substance and drug product so that we could be a one-stop shop given some of the difficulties that are associated with sourcing capabilities in this area. Okay. So that's represented in this slide as well. And then off to the right, you can see a couple pictures. Inside our early phase facility, you can see our advanced mobile clean room technology that we use for the high containment, high segregation manufacturing. And then the inside of some of those as well. So we can really cover the roadmap across the life cycle of the program. So the main discussion points we want to hit today are some points together, and we're going to move quickly through this, and we can definitely have additional conversations after this, if need be, to delve into this with a little bit more depth as well in any of the particular areas that folks are interested in. So some key points to consider as have worked with a variety of these kinds of programs, and really the thing is we need to focus on the product life cycle. So early phase development and understanding your developmental phasing is really an important tension that's going to come up as you go along. So a common one that we see is where we see folks that really just need a proof of concept for their construct that may be at odds with the long-term needs of the program. So a critical area of that is you might be in an adherent cell line, for example, that you know for your indication that's really a no-go for commercial manufacturing as well. So there's the tension there. So we're in fact working on some suspension technology to basically take this tension, off the discussion point. The other thing to consider here is that, let's say manufacturing with adherent cells can be quite expensive, funky, you get low yields, et cetera. We're perfectly capable of, for instance, using hyperstacks quite successfully, et cetera. But it may even be that maybe investing a little bit in development early on can actually help decrease the cost of your clinical supply. So the next one is the market itself is quite different from what a lot of us grew up in, which is recombinant protein manufacturing. So when you're using the cure word, so we don't throw that around casually, but many of these products have the potential for that. You really have this large installed base that you have to work through in the beginning. So you need quite a lot of product in the beginning of this, and then you really drop down to kind of this annual maintenance production. And it has a totally different life cycle curve to to what most of the biological products today look like. And really what that tells you is whether you, we think that that is, lends itself to outsourced manufacturing more than internal. But even if you go with internal, let's say on a platform, what this is telling you is it's going to be multi-product. It may be sequentially multi-product, but that's something that definitely that control strategy needs to be built in from the beginning. And then the next thing is scale. So it seems perverse, but you actually might need a small-scale commercial capability, because for many of these products, you don't need very much product for commercial manufacturing. So really need to be set up to do commercial manufacturing across the scales that are out there. So obviously the key big obvious one is products are live, virus and cells. And these viruses are designed to infect human targets, and the genetic alterations may be permanent. This has some pretty big impacts on, let's say, EHS issues associated with manufacturing and handling these materials, and also choices that you make around the controls, and we'll go into that a little bit more. Another really critical one that differentiates is your adventitious agent control strategy. As many of these processes lack any dedicated control unit operations, okay? So vendor qualification, so you need to protect the process at its whole boundary. Your vendor qualification, your control strategy, and then your alignment between the EHS and quality risk controls. In some places, that also creates a little bit of tension for you. And then the last one is the challenges around procuring drug product, getting access into drug product facilities, what format are you going to use, and clearly it needs to be in an isolated system, given the nature of these type of products. So I would suggest that whether you do this with a model such as the one we put up here, or you have one of your own, you really need an integrated approach to managing this program and kind of a stage-gated process. It's really critical for us, for example, that we start and end with our facility and equipment in a neutral state. And what I mean by that is whatever the construct is that we're handling, whatever the environment may become exposed to that, or we're using single-use equipment, so there's no fluid path carryover, but we'll say environmentally, we need to make sure that we can demonstrate in a qualified way that we've returned all of that equipment back to a base state so that it can be used for the next program or the next type of construct as well. And so we need to integrate that in. The other thing we've got to integrate in is all of the environmental health and safety issues associated with handling this. And the flip side of that is that we have to ensure that those controls don't perversely induce some quality risk in another place. And that really requires an SME matrix of different functions. What's the output of that? Well, the output is really custom to each individual program. So there are classes of these constructs that are there, but given the individual nature of those and the hazard pathway that they might present to people, is it a contact hazard, is it mucous membranes, is it IV only, becomes real equipment. So you should have a set of equipment, a custom flow, custom room layout, custom grades, pressure cascades, air changes. Potentially, you may have to have different cleaning agents, et cetera, and really you need a cross-functional implementation group. So for this, knowledge about your product from an EHS perspective is really critical. So your preclinical data, your route, your hazard pathways to humans is really critical. And so we bring in a variety of folks as we do these types of assessments, and then we derive the controls, and then we do the training and notification to our operators as well. So the other thing that's really important is to handle risk to the community. So biosafety committee, communication, bring in thought leaders from the community to understand what's going on in your facility as well. It's important. So just a quick example, just an illustrative example to kind of deal with this. So you might have PPE that's required in addition to the GMP gowning, right? So that's got to meet your GMP requirements as well. It can't be particle generating, can't be contaminated, et cetera. And then just like any other thing like the room or the equipment, you also have to be able to sanitize and inert that equipment, in between uses. Otherwise, if it's not capable of that, then it becomes disposable to the process, to the product. So we have to do a very detailed risk assessment around all of the materials that are used, including the PPE. So another example here is where your engineering controls might induce some other risks is, for example, you're going to use a negative pressurization for the environment where you're doing this manufacturing. So that could bring contaminants in from the surrounding area. So we control that by design of the clean room, and in fact, doing closed manufacturing is pretty much the rule as well. So there's a couple of different strategies. And then, of course, we need to protect the product from the operators, because these cell lines are, in many cases, barely permissive to human viruses. And so we need to have a health monitoring process for the operators, et cetera. So as I mentioned before, process closure is really critical to manufacturing these types of products because of that tension. So we need to close the process and protect it from the operators and the environment in particular because of the lack of the adventitious control unit operation. But we also need to close and protect the operators away from these constructs. We have a saying that says, you get to go home the way you came in this morning. And my wife would prefer that I come home better than I was, so I added the phrase, for better or for worse, you're going to go home the way that you came in. And that's one of the real linchpins of that is around this closed processing. So we've been working on this for a number of years. We started developing it with our antibody manufacturing platform so that we could demonstrate that with products that are relatively low hazard to the technicians as well. So beyond that, we go into the facility design and control. So we have designed a risk zone for our facility, and we've designed and detailed out all of the controls which go towards handling the pathways of airborne, people, equipment, liquid and solid waste as well. So flows, advanced MCRs, et cetera. And then for the room where we actually do this type of manufacturing, this is an example of an advanced engineering control that you could bring into play. So you can actually think of these rooms as a piece of equipment. It's like an isolator that you can walk in, and they can actually pass a pressure decay test. And they're set up for a full VHP capability, redundant HEPA filtration, and they are actually welded out fully. So we have full containment before we actually put the coatings on inside the room to create containment. So if you'll get this presenter. So there's actually a slight ramp down in, so there's freeboard around the outside. So you are in effect walking in kind of a bathtub of containment. There's a spill response switch here, et cetera. So another area that's really critical here in terms of the product is, as I mentioned, we need to have a fully isolated capability for vialing the material. So as we vial the material also, there is risk for these constructs to get onto the outside of the container. So you need a full sanitization procedure for your containers as they come out of the filling environment before they come back into the surrounding environment. We don't want to propagate risk to the operators, and we certainly don't want to propagate risk to the healthcare providers that are handling these vials in hospital as well. So So for that reason, we've definitely invested in fully isolated filling with a VHP in, VHP out capability. So just as a quick conclusion here, I think we're just about out of time, is we really need to meet all of the requirements, and they need to align, and then many of them are their intention. So what that really means is that you need to take a risk-based approach to these engineering controls and operational procedures, right? It needs to be a matrix approach. And then we need this high containment, high ease of changeover designed in, okay? And then the last one is, how are we communicating with this, with clients, regulators, internally? And we've got to become facile with the language of risk and being able to clearly document hazard pathways and get them down on paper. And then we really need to be committed to following the output of those risk assessments and sticking with that. So anyway, I think I'd like to conclude there and take any questions.
Okay, great. So before we get started with the audience Q&A, we'd like to hear a little more about the work that Fujifilm, Diosynth, Biotechnologies has been doing with industry groups around this. Can you tell us what you've been working on and who you're working with?
Yeah, so that's a good question. So we work quite a lot with the BPOG organization. They have a cell and gene therapy track forming, and in fact, we're hosting the face-to-face meeting in November here, and that's going to include tours of the facility layout that we showed here. It's really critical for us to work with other folks who are working in this area. We're through that reaching out to the regulators, and so this emerging area, is going to be it's going to need development and focus and thought, and we don't have a 20 or 30 year history in handling these materials, so we definitely need to all work together to to push these life-changing materials forward into the market.
Okay. And so we do have some questions from the audience. So as a reminder to the audience, go ahead and type those questions into the chat window. So the first question is, do the EHS considerations only apply in the presence of a virus?
So no. So some of the cell lines that are used in here may be derived from other mammals. There may be issues there, so we need to understand any of the EHS issues that arise from even the cell lines that we're handling. And then we're certainly doing an enormous amount of characterization around those, even the cell lines, before we bring them in, because of the requirements for the adventitious control are incredibly elevated with this. So you could apply some of these same constructs to handling microbial or other types of systems as well. I would just say this space just requires you to take it that much further and be that much more meticulous.
Okay, great. So thank you, Thomas.
Thank you.
Thanks to our audience for joining us. If we didn't get to your question today, we're going to pass it along to Thomas, and he'll be able to follow up with you directly. The recorded version of this webcast will be available for on-demand viewing on our website. As a registered attendee, you'll receive a follow-up email providing you with a direct link. We look forward to having you join us at future Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.